Okay, hello everybody. This is Psych 3280 Neural-Based Consciousness Week 2. And this week we are going to talk about introduction to the research methods. So before starting, a quick recap of what we did in last week. I just talked about the global organization of this lecture series, explaining the global learning objectives, and also assessments, and also there, I asked you to go through why, and then what, and how, when you're making some kind of presentation, general club presentation, as well as writing, which I'm going to explain to this week. And then I also talked about the global view of what kind of papers are available for assessment to general club presentation. And then as an optional, I also gave a brief presentation on how to effectively read books and papers. And then in terms of the lectures, I also discussed about the problem of consciousness, studying consciousness. What does it mean to study it scientifically? Because some people may believe that the consciousness is a, subjective thing, and as Galileo said, it's never be a scientific target of the study. But that's not the case. That's what I'm trying to convince you throughout these lectures. And then as a sort of a critical backbone of this lecture series, I explained phenomenology, experimental data theory, triad. And we are going to talk about it today as well. And the impacts of consciousness research, mainly clinical, but also there are lots of issues that benefits from consciousness research. Okay. And then difficulties in the study of consciousness that we face only sort of the particularly for consciousness, but not other kind of the behaviorally observable stuffs related to the brains. And then at the end, I introduced a little bit of the anatomy and so the actual research in consciousness research. So this week, we are going to extend this, and you'll be able to think and answer about the following questions. First, how can we characterize our own phenomenology? So the method-wise, we extend it. And what do you see in the moment? You might have thought about this before by yourself, by blinking on and off like this, and then try to see how much you can see in a moment. And if you haven't, maybe you can do it right now. And there's lots of questions regarding to this, related to the richness or sparseness of the consciousness. And then third is, as a sort of extension of this second question, what is the phenomenology of so-called backward masking? And this technique, backward masking, is a popular technique in consciousness research, so I'll give you a demonstration for this. And then how can we quantify the degree of conscious seeing? So this becomes much clearer once you experience by yourself what the backward masking is like. And then, is it actually possible to quantify this subjectivity? That's the question. And then the latter half of this week, I'm going to explain what are the methods available to measure neuron activity in the brain to promote consciousness research, and what would be the required kind of assumptions for these methods. And then in particular, we are going to talk about the two types of the neural measures. One is EEG, and how EEG is generated by the brain. That's the question. And also, we are going to talk about fMRI. That's another non-invasive imaging technique, and we are going to discuss about the source of these two signals. And as a reading material, I suggest Quest for Consciousness chapter 2.